All right, well, welcome everyone. I'm Amy Franco. So just for those of you who haven't seen me or been to one of my sessions earlier, I am a scientist. I'm not a clinician, but I'm a scientist at the Children's Hospital of Philadelphia. I'm also a 25-year follicular thyroid cancer survivor. So I was diagnosed with an FTC during my senior year of college. It kind of put me on a path a little bit towards research. I had always thought I was going to be an MD because I was that science geek growing up and I loved science. And then yeah, this is us. And I always thought I would be an MD, and then I realized MDs didn't do the fun stuff that I like to do about science, the discovery, the experiments, and things like that. And kind of through my own diagnosis, I realized there existed this entire career to do the research aspect of it. I will say I never dreamt I would be back in the thyroid cancer space. I did a PhD at Vanderbilt, and I was looking at stomach cancer and bacterial infections and how H. pylori increases the risk of stomach cancer. And then when I did my postdoctoral training, I actually ended up at Memorial Sloan Kettering and ended up in the thyroid field and have not left. So I started my own research lab, and I was at the University of Arkansas for Medical Sciences in Little Rock, Arkansas for 8 years, focused primarily on adult thyroid cancers. Did a lot of work with mouse models, cell lines, trying to really understand the genetics of thyroid cancer and what was going wrong. And it was during that time I started talking with clinicians and realizing one of the deficits in the field was understanding the differences between pediatric and adult thyroid cancer, both how they behave, the genetics, all of that. And I had the opportunity to move my lab to the Children's Hospital of Philadelphia, and that's where I am now. And we're doing a lot of the work we were doing in the adult space, but really trying to understand the differences between pediatric and adult thyroid cancers and why they act differently. So that's a little bit about myself. Today's talk is we're going to be talking about genetic and molecular testing. I guess the first thing I'm going to say is who's had genetic testing or molecular testing? Okay, good, good. That's great. Who knows what molecular genetic testing is? Okay, good, good. That's perfect. I wanted to get a little bit of an idea. So we're gonna introduce and talk a little bit about genetic molecular tests, what it tells us, what it doesn't tell us, and we're gonna go a little bit into the difference between somatic testing, so when we test the tumor, versus germline testing, which means something is wrong in the germline in all cells of your body and how that may predispose to cancer. So first what are molecular tests, right? So when we talk about molecular tests and we throw around that buzzword, we're most often talking about things like Afirma, ThyroSeq, ThyGenX, Thyromere. These are some of the commercial tests that you might hear FNA is sent to. But many institutions also have their own molecular tests. And CHOP, we have a CHOP comprehensive genetic panel. It really depends on where you're at and what's available, but these are the three most common commercial tests. So especially if you're not in a large center, and center of excellence or at a cancer center that has a real high volume, that has its own personalized test, MD Anderson, CHOP, Penn, Harvard, they have their own individual tests, but otherwise it would usually be sent to one of these commercial tests. And that's what we talk about with the molecular tests usually when we're talking about testing a tumor. At FNA, trying to use that to rule in or rule out thyroid cancer. It comes into play when we start to think about whether we can use molecularly targeted therapies. That means using a specific drug or therapy specific to whatever changes is happening within your own tumor. So we really try to tailor it, or as we call it, precision medicine, to what your tumor is. This is different than genetic testing. All of it is looking at genes, looking at the genetic code. But when we talk about genetic testing, that's when we're usually looking at things that are in the germline. Sometimes genetic testing will be 23andMe. You can get all of these ancestry type of tests, which a lot of time, again, look at your genetic code, and that looks at the DNA that is the blueprint for everything, but it's not specific necessarily to a certain organ, the thyroid, the lung, whatnot. It's what's in every single cell. So that's where I get into this germline versus somatic testing. And I'm sorry, this is a really small screen. Despite my glasses, I can't see it. But when we talk about germline testing or germline mutations, what we're thinking about is something that got transcribed and is in your genetic code that every single cell in your body gets. There are caveats to it, but just think of it that way. That's the blueprint. That's your DNA that goes into everything. And you also have the potential through sperm, egg to pass that on to a next generation. So those are our germline mutations. Then we have what we call our somatic mutations, or those are the mutations that are specific to the tumor. That means only the cells, in our case, only the thyroid cells have that mutation, that irregular piece of DNA. We don't have the ability, if it's just in those tumor cells, to pass it on to our children, to the next generation, because it's encoded and it's very unique just to our tumor, not to all of the DNA that gets passed on to our offspring. So it's only tumor cells that have it. And I think that's what's important. And I think often we don't communicate that. And we, meaning physicians and the medical community, of communicating whether we're testing just what's in the tumor versus what is a heritable or what's a germline that we have the possibility of, one, inheriting from our parents, and two, potentially passing down to our children. So germline testing, I think we've started to talk about this more in the context of thyroid cancer, especially over probably the last 10 years. And actually, we were talking about it just recently in that roundtable. Some of us were talking about, some of you made it or didn't see it in time, that was at earlier today. But germline testing, we do know some things that are associated with thyroid cancer, Dicer1. We don't need to go into all of the specifics, and we can talk in question and answer if people are more curious about some of these specific mutations. But we know that these syndromes, like I said, Dicer1 mutations, P10, MEN2 with medullary thyroid cancer, those can be germline mutations that we can pass on in families and we know are associated with thyroid cancer or an increased risk. It doesn't mean that you're going to get thyroid cancer with some of these mutations, but it does mean that you have an increased risk. But the more research we do and the more that we look into this, we actually understand that there's probably a whole spectrum of other germline changes and mutations that you may inherit that could be associated with an increased risk for thyroid cancer. But if you don't look, you don't find anything. And in the past, a lot of these studies looking at big cohorts of individuals and looking across populations weren't necessarily looking for associations with some of these changes with thyroid cancer. So the further we get along in understanding the risk and the more that we look at families and seeing different types of thyroid cancer that actually seem to be passed on between families, we're now starting to get a better idea of even more things that fall into these categories and mutations or changes that may be associated with an increased risk of thyroid cancer. But again, I think it's really, really important to understand is that these are associated with risks. These are not associated with promises or guarantees of thyroid cancer. And my own personal bias is I think anyone who's using germline testing and looking at some of these changes that happen in the germline really need to be doing this quite closely with a genetic counselor, with a partner physician to really understand what are the risks. Sometimes, knowledge is power, but also too much knowledge with not knowing how to apply it can be extremely dangerous as well. And I think it's really important to understand that these changes are associated with risks. FAP, which we know is associated and really a predisposition for developing colon cancer, the lifetime risk is only an increased 2% risk in thyroid cancer. So it's not a panic factor that I'm going to die of thyroid cancer if I have these, but it definitely means that you want to have that increased awareness. And absolutely, as you age and knowing that you potentially have this risk, is to look for thyroid cancer. But again, knowledge is power to be aware, but not necessarily, like I said, it's not a guarantee of something is going to happen. And So going into this is, what if other family members have had thyroid cancer? We now are seeing more families where we're seeing the association and multi-generations have developed thyroid cancer. We historically had kind of said that was crap luck and that there wasn't necessarily a genetic or a germline uh, tracking of this, but I think now we're getting a much better idea. The deeper we look, the more sequencing we look for, that there are definitely what we call risk alleles or factors that we haven't figured out. One of these is SNPs, and you'll hear it in what it is small nuclear polymorphisms. What it means is that when you have these changes, they can be associated with an increased risk. We don't even know what all of these things do, whether they lead to increased expression of some proteins or genes. We don't understand really that full risk. But the more we look, the more we're starting to see the correlation between certain changes and a risk for thyroid cancer. But what I'm going to say is correlation doesn't mean causation. And again, it's just these increased risks. So this is why I think, especially when we're talking about germline testing and we're thinking about things that are passed between parent to children and across generations, that it's really important that this is done with a medical professional to really set the stage to understand both the pros and cons of the testing, but then also understand what those risks are that are associated with it. And when a change needs to be taken, when is it, with MEN2 mutations, when does it mean that we need to prophylactically be concerned about medullary thyroid cancer and have those patients undergo a thyroidectomy because we know they're at such risk of it, versus having FAP, which has a 2% lifetime risk? I certainly would not recommend that you go and get a thyroidectomy on that risk. So we need to really work with professionals that understand this, and especially genetic counselors. Who should be tested? This is a conversation that I can't give you that answer, but if you know that there's multi-generational incidence of thyroid cancer within your family, it's probably a really good idea to start talking and bringing that up to your clinician, your endocrinologist, and really start to think about whether it is appropriate to have germline testing. And it's not going to guarantee you're going to find something now, but who knows, in 5 years, we may know more about some of those changes and find some of those associations. So that's talking about the germline testing and talking about the mutations, those changes that we see that all of us carry, that it's in every single cell of the body. But as you saw from that first picture, like the FAP, which I'll just use that as an example, it is not a 2% lifetime risk for developing colon cancer. It's a 2% lifetime risk for developing thyroid cancer. We still don't understand why these changes, even though every cell in the body has them, why some organs are more sensitive to those changes than others. There's still so much that we have to learn. So that covered germline testing, but more commonly what we think of in the context of thyroid cancer, and I will say germline testing, most of those mutations that we talked about, we're going to talk a little bit about oncogenes versus tumor suppressor, but those mutations that we found in the germline, those genetic mutations confer risk. Most often, that doesn't mean that they have something that's targetable, meaning identification of those doesn't mean that you can use this drug. When we start talking about molecular testing, we're starting to look at refining our diagnosis, but also trying to use those changes to identify whether we may have a new drug or a new intervention that'll work and only will work if you harbor that. An example is, probably many of you have heard of BRAF. If you have a BRAF mutation in V600E, we have very specific drugs to treat that. But if you don't have a BRAF V600E mutation and you go on those drugs, it could actually do more harm than good. So that's why it's really important to kind of know the distinction. So molecular testing, we're going to go through the different phases of when it can be utilized. But molecular testing more often has the ability to help us identify potential new therapies that could be utilized. That's not the same case with the genetic testing. The genetic germline testing is really to identify those patients that may be at risk. So Molecular testing. So many of you, I mean, we already talked, but I don't know online because I can't see hands, may have had this done with an FNA. So at first we were using molecular testing to really try to distinguish and refine our diagnosis of thyroid cancer. And we knew that taking the cells on an FNA, looking at them under the microscope, there were some that were always very obviously thyroid cancer. We said, yes, this is thyroid cancer. We sent a patient to surgery. However, there's a lot of those cells that come back and either there's not enough cells that were collected or they look funny, but we're not sure if they're cancer. Or in my case, I had follicular thyroid cancer. You can't diagnose a follicular thyroid cancer until after you take the thyroid out and you look at the pathology. It's just not possible to diagnose only by an FNA. So for that, we really started to develop these molecular tests to use at FNA at the time of biopsy to try to see whether there were genetic or different changes that were happening that we could then say, yep, this is a high probability of malignancy, intermediate probability, or this is a low probability. And it was really important for us to have what we call the negative predicted value, where we wanted to really clearly figure out which tumors that the cells looked a little funky, but they weren't cancer. And if we could find something from the molecular test that said, this is indeed not going to be cancer, and we have a very high level of confidence on that, that could save a lot of patients from undergoing a thyroidectomy or a lobectomy just to find out it was not a cancer and decrease quality of life that follows with it. So like I said, this is really, when we do this type of testing, we're only looking for changes that are occurring in the tumor cells themselves. And this is why. I mean, we already went through it and I talked about it, is, when we do an FNA, we get the cells and then often we can say, yes, indeed, we think it's papillary, we think it's a tumor, yes, it needs to come out. But too often we end up with these gray areas and we don't know what to do with it. And that's why we developed, or we collectively, the community, these molecular testing panels. So again, why not everyone gets it is if it's a very cut and dry, clear case that this is definitively papillary thyroid cancer, definitively an ATC, well, that's another case, but we may not need that molecular test as well if we already have that positive diagnosis. And you can just see this is just some of the panels. But we've now also gotten to the point with some of these panels that not only are we looking for some of the mutations that we know are most commonly associated with thyroid cancer, BRAF, RAS mutations, some of these fusions, RET mutations, we also are starting to look at some what we call gene expression or microRNAs. And we can see, are there different genes in that tumor that are up or down that really give us a better idea of, yes, it is cancer, or no, it's not. So we've really advanced the field in many respects to not only look for just mutations that are associated with cancer, but what we call them now are signatures and gene expression signatures that may be associated with cancer. So what drives thyroid cancer? And I put this up here. This is your little biology for the day, but you don't need to remember it. But what we know for thyroid cancer is that the vast majority of these tumors are driven by what we call the MAP kinase pathway. This is a pathway that is absolutely critical for all of our cells to grow, divide, to survive. In thyroid cancer, it goes wonky and it can't stop. And that's why the thyroid then grows and becomes a tumor. This is an essential pathway. Every single one of your cells is dependent upon using this same pathway. It's just the difference is usually every other cell without a mutation, without some of these changes, can turn it off when it needs to. In thyroid cancer, the problem is it doesn't get turned off. And that's why you end up with a tumor that progresses to thyroid cancer. But this just shows kind of some of the most common mutations. And this, I'm going to caveat, is in the adult setting. Because we know that there are now a lot of subtle differences between pediatric versus the adult tumors. But for the most part, with follicular thyroid cancers, we know that they have these P10 mutations. That was one of those germline mutations as well that I told you about, that it can increase risk. These PPAR gamma PAX8 rearrangements, so this is when two genes get stuck together when they're not supposed to be and they get turned on. RAS mutations. And then in the context of PTC, Papillary, we see BRAF mutations, these RET fusion rearrangements, and then anaplastic usually have both the mutations, but then they start to acquire more things that go wrong so that those cells just can't stop themselves. And that's what gets us to what are the different types of mutations that you see? Are they tumor suppressors or are they oncogenes? And I don't want to go into all of the biology and overwhelm you, but I think it's really important when we think about cancer, we think about tumor suppressor genes, and you can think of those as the brakes on the car. They're what control us. So if you lose one brake on your car, you got a problem, but you still have the emergency brake that you can pull, and you can usually come to a stop. It's not going to be coming quite as quickly. So with tumor suppressors, there's two of them. You need to really lose both of them. You need to lose both your brake and your parking brake before you're going driving off the cliff. So that's what tumor suppressors are. But those are also the genes that we often see in those germline predisposition. It's tumor suppressors. It means one copy isn't working right. Those brakes are not the way they're supposed to. They're not functioning properly. That's why you have the increased risk of thyroid cancer or any other cancer with those germline. The other part are the oncogenes. Oncogenes, you can think of those as the accelerator. Once that accelerator gets pushed, you're in trouble. The oncogenes, all it takes is one copy of those, one change to happen to get that accelerator stuck. So you only need that to happen once, the oncogene, and that's what accelerates. That's when you hear about BRAF V600E mutation. Just one mutant copy of that puts on the accelerator, and that's when you end up with a cancer. So this is just, the idea is to kind of think about this to understand. And when you see some of these molecular changes, the molecular tests, one mutant copy of PTEN, which is a tumor suppressor, increases the risk and it can cooperate, but in and of itself, only one copy that's wrong isn't going to set you off the cliff probably into cancer progression. So that's why, and I'm just going to go back. So this is when we're talking and we're utilizing these molecular tests, I should have more clearly talked about it. When we talk about molecular tests, at the time of FNA, we're doing it really to try to get and refine that diagnosis. Is it cancer? Is it not? Hopefully, we get some information out of that, that if it is indeed cancer, and it turns out that that cancer has spread, metastasized, surgery isn't enough, that we need something else beyond that for therapy, we hope that that molecular test has identified something like BRAF, where we know now there's a new drug that we can use in response. Identified RET, we have lots of RET drugs. NTRK fusions, we have specific drugs that target those kind of changes. But the reality is most often when we're doing that at the FNA level, we're really looking to refine the cancer diagnosis. But it becomes really important to understand, is it in these tumor suppressor? Is it in these oncogenes? Because on that token, if thyroidectomy isn't enough, if RAI isn't enough, if we have persistent disease or we have metastasis that can't be surgically removed, we may need in our arsenal different drugs. And that's when it becomes really important to have some of these molecular tests and to understand what these genetic changes, what has happened. Because fortunately, we've learned from other cancers that these different mutations and these different changes can be targeted very specifically with many other types of drugs that are available to the cancer community at large. It's fairly new that we've started using these so aggressively and so successfully in thyroid cancer, definitely since the time of my diagnosis 25 years ago, where we've really understood that we, with persistent, with resistant disease, sometimes with recurrent disease, that we can use these different types of drugs. But this is when it becomes very important after FNA or after the time of diagnosis to have the options for some of these molecular tests so that we can identify these specific changes and whether there is a drug that can fit into this paradigm or that can specifically inhibit or work on those mutations. And I'll bring up BRAF because I think it's a really important example, is we have some really good targets and drugs that are available to BRAF specific. But if it turns out you had a RAS mutation, not a BRAF mutation, and you go on one of these BRAF drugs, it will super accelerate that cancer. So it's really important for us to open up the opportunities for some of these very specific drugs, is to know what kind of mutations are available, or what kind of mutations are there within the tumor. The biggest problem is that we're way behind in thyroid cancer. This has been done for decades in other cancers. But most often when we come in the door as thyroid cancer, we come in through oncology, or excuse me, we come in through endocrinology and through our surgeons, not through oncology. So there's really a big divide between what's available, and it's also further exacerbated when you're seen at a center of excellence or a high volume center or one of the cancer centers versus being seen in the community center or being seen in the community setting. So I think, it's really important, and this is an absolute game changer for many thyroid cancer patients with persistent disease, with metastatic disease, that they absolutely can benefit from having the molecular testing. There are also a huge number of thyroid cancer patients that having the molecular testing will never change the course of their treatment. If they have a small microcarcinoma, many of them will do very well, or having an isolated FTC only in one lobe will do very well with a lobectomy and with standard of care. And it's not necessarily imperative having that molecular test is going to change the treatment paradigm. So I think this is where I bring this up, that molecular testing, germline testing is not a crystal ball. It's not going to give all of the answers. Knowledge is power, but each of us is different and each of us is unique. And not every individual is going to benefit potentially from molecular tests. And not every physician, unfortunately, and every endocrinologist out there is going to know how to use this testing. So I think this is where it's really important for us to be our own advocates. It's really important for us to ask questions, push a little bit, as difficult as it is, and know that it's okay to fire your doctor if you're not getting the care and moving forward in the way that you need to. That it really is important for us to continue to ask questions. But like I said, it isn't a crystal ball. Molecular testing isn't the panacea to give us all of the answers. Genetic testing isn't going to give us all of the answers. And I think we have to be cognizant, because knowledge is power, but too much of a good thing also can be a bad thing at times. And to not understand what exactly the molecular test can and can't tell us, telling us risk, but not guaranteeing malignancy. You need to be able to have those conversations with a physician, and with, I think, with ourselves, and be empowered to have that. Is the 2% risk of, with FAP, for example, that 2% risk of getting thyroid cancer, for us, is that worth the long-term potential side effects of having a thyroid removal on a 2% risk, or not? And for some of us, that is going to be worth it, because that 2% risk is going to keep us up and we're never going to sleep soundly at night thinking about it. Whereas for others, it is going to be, I'll take that 2% risk. We'll go in for ultrasounds yearly, and I'm just going to be aware and make sure that I told my physician they need to palpate my neck, they need to do ultrasounds, we need to work for it. So that's where it becomes very individualized, and important for every individual to know what is right for them is not going to be right for the person next to you. And I hope through this and through these conversations and having ThyCa and things like that, is to understand there isn't going to be a one size fits all that's going to work for everybody, and to understand the pros and cons of having some of this additional knowledge. And at the end of the day, tumors are really complicated. So we talked about the mutations that happen within the tumor cells, but then those changes that happen with the tumor make it act differently and talk differently to all of the cells that are around it, be it the immune cells, be it the blood vessels. So tumors are really complicated. And again, it becomes very important to understand what we're looking for, what these tests tell us, but also to understand what they don't tell us. And they don't tell us everything. So that was kind of the broad overview. I ran through this really quick, and I apologize for that, but I hope that we can open it up to conversation and questions and maybe moderate. I know we have some online, and I'm happy to go back because I know some people were taking pictures of slides and things like that. If there are things that we want to go back to that weren't clear or you want more detail on, let's do that.
Can you talk to me about where oncocytic cancer is in some of the slides?
So yeah, oncocytics. So they don't fit into any of these paradigms.
Thank you.
And that is the exact challenge, right? We've gotten really good for papillary thyroid cancers. That's what's been studied the most, and understanding a lot of the mutations and the changes that happen in papillary, and identified BRAF, RET fusion, some of these that are really the most well known. But when we start to get into these smaller subtypes of thyroid cancer, our knowledge is a lot less refined. And these smaller subtypes used to be bucketed into other cancers. So 25 years ago, oncocytic was follicular, right? It was a subtype of follicular thyroid cancer, and we thought it had all of the same mutations as follicular thyroid cancer. Then as we started looking more, we realized, yeah, no, they're not the same thing. And it turns out they don't even have all the same mutations. And actually, we just published a paper that got accepted last week on oncocytic thyroid cancer. And what we were looking at is understanding in the tumor microenvironment, that's where oncocytics are so different. They actually have different crosstalk between the macrophages, so immune cells, and the way they talk to the thyroid cancer cells is very different. And we actually did a comparison because historically oncocytics were thought to be like a follicular thyroid cancer. So we compared follicular thyroid cancers with oncocytic thyroid cancers. And again, where it's ever changing is when we started this project, they were Hürthle cell cancers. Now they're oncocytics. So we're always learning and we're always refining some of this knowledge. But as it turns out, the communication between oncocytic and the tumor microenvironment, those other cell types, is different. The other thing that is very different about oncocytic thyroid cancers is their metabolism and what we call their mitochondria. Again, I don't want to overwhelm with the biology, but the energetics and the way those cells just produce energy and respond is very different, and we don't understand why. So we do know that they have what we call mutations within their mitochondrial genome. So that's not even anything that's tested on any of your molecular tests, not tested when you look at germline. Biology is really complicated. And we tried, I think, initially in thyroid cancer to simplify things, and that worked for a lot, but it didn't work for any of the subtypes. And even when we started to get into anaplastic, it doesn't follow the rules that anything else does. So I think, thyroid cancer in many respects is still in its infancy in the research aspect of it, because there's still so much that we don't understand. We do well for, I would say, 75% of patients whose tumors follow the rules that we established and we thought thyroid cancer was. But that 25%, and I'm making that number up, but I think it's pretty accurate, that don't follow the rules, the oncocytics, the anaplastics, the follicular thyroid cancers, those smaller subtypes that just fit into a different bucket, we're still trying to figure it out. The problem is it's seen by our endocrinologists. We don't have the same level of research for thyroid cancer that we do for other cancers, because I've never had a thyroid cancer class in any of my cancer biology. No one talked about thyroid cancer in cancer biology. That was the other tumor, and it gets seen somewhere else and it doesn't come to a cancer center. So there aren't a lot of us out there that are doing this. And there is this, we have fantastic clinicians, but they don't ask questions the same way or do research the same way that I do in a lab. They don't have models. So there's that disconnect, and that's where we're still trying to figure it out. That was a very long-winded answer, but but oncocytic just doesn't follow the same rules, and it has RAS mutations, but those RAS mutations do different things, it seems, in oncocytic tumors than it does in traditional follicular thyroid cancers. And a lot of it probably is related, like I said, to these differences in mitochondrial genes and how these cells produce energy and how they work. It probably, you can, but I'm not sure that it would tell you anything because we don't, in the oncocytic, there may be, again, in the very small, you may be the unicorn within an oncocytic subtype that has a mutation that would come into one of these that has a molecularly, as we say, targeted therapy. But the reality is to date, we haven't found any of these most common players that I would say universally or in most oncocytic, and we don't have the specific drugs yet for those mitochondrial changes and those energy changes yet, unfortunately. But from research, yes, get it, because the more data we have, the more we can find and ask these questions, I think.
So those adjunct medications maybe wouldn't work? Do you see what I'm asking?
The what?
Those are medications,
Yep,
right?
each of these are drugs
Yes,
that
right.
target. So if an oncocytic tumor doesn't have a RET mutation, none of these drugs are going to work.
Right. So it's just they make a guess at which one might work.
Correct. And that's where some of these, so selumetinib, trametinib, these are drugs that are that hit many different types of mutations. So they're a little bit broader. So like a MEK inhibitor could actually work for anyone that's got something that's further up than that, and it may work. However, it's not going to work with somebody that's got something below it or over on the other side. That's kind of, and these very specific BRAF mutation inhibitors, those only work if you have that very specific mutation. If you don't, they won't work at all. So that's where, the pros and cons, but looking for oncocytic when it's not in one of, most oncocytics aren't driven that we know and have these mutations, that even having it isn't going to open up an opportunity to have one of these drugs because chances are those mutations aren't going to be there. It's going to be something we haven't found yet.
And we have one question online
And they'll come
from
here.
Catherine with a follow-up. So if I had Thyrosec molecular testing 10 years ago, is there any benefit to retesting if there is recurrent disease? Followed up by, also, what does it mean if a report says I have a 7% allele frequency of BRAF V600, no other mutations or gene fusions?
Right. So the different molecular tests, they will tell you what they've tested for and whether it's there. So allele frequency means when they tested, like how prevalent was that mutation? Did they find it in most of the cells, only in a few of the cells? That could mean that one, there wasn't enough cells in the sample that we didn't get an accurate measurement, or it could mean that it was only in 1% of the cells, so we really don't know if it's actually that important or not. And I have to say, I don't do the clinical stratification based on the molecular tests. So this is where it is so important to get the reports, to read the entire report, but then also to talk with your physician. And the reality is that, I'll go back to, it's not a crystal ball. And we're trying, all that we report is based on what we know from the past, but not what we can predict from the future in the future. So with your kind of related to the question about is it beneficial to have another test? Yes and no. If going to this, often when we see progression of disease, you have a mutation in the oncogene, puts the accelerator on, but as it progresses and maybe when it goes to the lung or it comes back, it's actually lost the brakes. So now it's got a second or a third, we call them hits, a different mutation. So molecular tests may tell you that, but not always. And the other thing I will say is these molecular tests are also constantly changing. So they're adding new mutations. So it could be that they didn't find a mutation when you were tested 10 years ago because we didn't know it was important. It wasn't on the test, it wasn't on the panel. But now with what we've gained from knowledge over the last 10 years, it may be that those mutations are there. And I'll give you an example for in the pediatric setting, we know that we call them kinase fusions. Those fusions, two genes get stuck together and get turned on when they're not supposed to be. We never used to test for NTRK fusions because NTRK wasn't common in adults. It didn't make sense to put it on these panels because more often than not, you weren't going to find it. So we didn't know that it was actually present in pediatrics because it was never detected because we never looked for it. Now that we know it's so common in pediatrics, we're starting to look more, and now we find out that it's more prevalent than we thought it was. So there is absolutely some utility to repeat testing on some of these cases because we know more now than we did yesterday. I know we have a question here.
I think my question's a very nuanced question. And it's more out of curiosity of upcoming research. The connection to PFAS chemicals and whether or not there's any research that's going to take place or is already taking place on that driving certain mutations or even the reverse, certain mutations being more receptive to those chemicals.
So the forever chemicals, yes. So it is pretty nuanced. And I will say, so we don't know whether, I mean, at this point, we don't know specifically whether it's driving certain mutations. We tend to think probably not, just because at least not in specific cells. However, what we are concerned about is kind of going back to that allele frequency question of if you only have 2 or 3 cells that might have a mutation in it, and it was just going to stay there with those 2 or 3 cells, and it was never going to develop into a cancer. If you're exposed to these forever chemicals, PFAS, does it all of a sudden, those 2 cells become 4 cells, become 8 cells, and they blow up into a tumor much faster than they ever would have had you not had them? Those are really difficult studies to do, especially when we start talking about population science and talking about maybe it only affects 1% of people. That makes it absolutely significant for that 1%, but it makes it really difficult to find it on a population-based level. So we are absolutely asking those questions. It's funny you bring that up because I've just started working with a group at the University of Colorado and trying to figure out, 1, how do we ask that question? And 2, how can we use, and I'm working with epidemiologists, those that study populations, but we're also brainstorming then how can we take that question into the lab and kind of artificially mimic it? Can we use mouse models? Can we use cell lines? Can we take cancer cells that have the mutations and expose them to the PFAS, to these forever chemicals, and figure out how those chemicals specifically change tumor cells versus normal cells? So we're absolutely starting to ask those questions. The problem is 1, there's not enough research funding dollars, and 2, there's not enough people in this field. But we definitely are trying, and those are the types of questions that I think are going to be so impactful but this is emerging science. We would have never even talked about this 10 years ago. And are we too late to be talking about it now because the forever chemicals are in everything that we eat and everything that we drink? Is it, well, not too late, but how do we fix it? So yeah. And the other thing about PFAS too is that we know that they have the potential for being endocrine disrupting chemicals. We call them endocrine disruptors. So we know it changes the biology and how a thyroid cell works, how it makes thyroid hormone. It changes its normal function, but how that impacts whether it will become a cancer versus not functioning right and leading to not producing thyroid hormone appropriately and things like that. Those we don't know the answers to those questions yet.
There's no more questions online at the moment. So why wouldn't she push for it? Yeah.
So why wouldn't our new friend Cora, right, push for, to get the genomic testing, because maybe she'll be ahead of the time, they'll find something.
Right.
And then if more,
Yeah, no, you're good.
yeah, I think that maybe the more people with oncocytic, thyroid cancer will get tested, and they'll say, hey, look, this is what we're finding.
Yep.
Someone's got to start it somewhere.
No, you're exactly right. And I think, and this is where the challenge comes between what we do with research and what we do with clinical practice. And they're not completely aligned. Am I still on? You are? Okay. And I absolutely say with 100%, everyone should be tested. It's not financially feasible. And where does that information go? Because if it doesn't go into a database where I can look at it, where I'm putting all of this information together, and instead it just sits in your chart and your physician doesn't know what to do with it, doesn't publish it, doesn't let it go to the broader community. And selfishly, I say broader community, me and other investigators that are looking for these associations, that's where we have the problem. So this is where, and I'll put on my patient hat, where we as patients have the ability to really push for decentralization of this data. How do we preserve our own privacy and ensure that our medical records aren't grossly released, but at the same time making sure that this information, if there's genetic testing that's done, that it becomes widely available to the research community, because your physician, and I have no idea, but I can say maybe they see 5 oncocytic in their entire career, and all 5 of them have different mutations or have different changes. That doesn't get us closer to the answer. But when we take that information and go with 300 other physicians that each have 5 patients, and then put that data together, that information together collectively, that's when we can start making a difference. The problem is right now is too much of this is decentralized, and it's in so many silos and pockets. And again, I'll come back to, we're not seen most oftenly in oncology, in these cancer centers, part of these large biorepositories, part of these large databases that have all of this information together, that it's really hindering us from making progress and understanding this. We learned a lot research-wise from what we called the Cancer Genome Atlas. So these were initiatives at the federal level to look at mutations that are common in different types of cancer. I still to this day have no idea how they ever got it approved to have it done in thyroid cancer, because we are so grossly underfunded and don't get anything. But they did. They have it for breast, they have it, and we had 300 to 500 patients that were enrolled in this, that they studied their tumors and they did all of the molecular testing. And actually, it wasn't targeted molecular testing, it was whole genome. They looked for everything. Problem was, it was done on papillary thyroid cancer, which is great because that is the most common type of thyroid cancer. And there were some that were mixed in, follicular variant of papillary thyroid cancer. But it didn't do anything for the rest of us that have a different subtype, and we didn't get that information. So, we still have a long ways to go to try to get all of that information together. And I think this is where we have a voice and power as patients to continue to demand that and to continue to ask for it. And ThyCa actually has done a great job with the research grants and administering them and funding specifically thyroid cancer research, because it is so difficult for us to get funding anywhere else. There is so little federal dollars and other institutes that invest in thyroid cancer research, unfortunately, because they say 95% of your patients don't die, so we don't need to know anymore. And yeah, well, that's great. There's a hell of a lot more to life than living and dying. There's a whole roller coaster in between that we want to enjoy. And, I think we have to change the narrative as patients, but also as researchers. And this has really been my passion and what I do. And I think having a unique perspective of researching in this field, also having lived through this diagnosis, that we have to change the narratives. It's not just about living, it's also about thriving and understanding how can we further refine our care so that it's not just looking at a yes, no, are they dead, or are they alive? There's too much more to ask and so many more questions. And if they want to say, we don't die of our disease, half of these mutations kill patients that have these exact same mutations in other organs. Well, then let's optimize and say, what's different about the thyroid? Let's learn something and figure out why a BRAF mutation isn't a death sentence in the thyroid, but it could be a death sentence in a melanoma. Why does it act so differently in a thyroid than in a melanoma or in a skin cell? So I think those are ways that, I try to spin the narrative as a researcher and just try to figure this out, because there is so much that we can still continue to learn from this disease, from the biology, refine how we care for patients, not go in there with, a huge thyroidectomy for a patient that may be just fine with a lobectomy. And how do we refine that? And alternatively, we know ATC is practically a death sentence. And we have a lot of research. And if you're a center of excellence and you're an MD, fortunate enough to be at an MD Anderson or a Sloan Kettering, there's a lot of opportunities. But those opportunities don't exist in all of the community centers and don't exist at all cancer centers around the country. So the disparities that exist in how and where you're treated. I was diagnosed in a community center. They would have never even thought about doing a molecular test. And to this day, they still don't think about doing a molecular test unless they need it to rule in or rule out thyroid cancer. But they're not doing it for the reasons that we talked about for refining treatment or opening doors for having other drugs available.
So I have two daughters in their 30s, and when they go to their primary care doctor and are asked questions about family history, they say that my mom, meaning me, had breast cancer when I was 47 years old. So then they will now get mammograms earlier. And they also say that I have this oncocytic cancer. And then one of my daughters' physician found the Cowden's disease. And so what do I do? How could I help my daughters? Like, what kind of testing should I have that would give them information for their future?
So Cowden's disease right here, that's your predisposition. That's your P10. That's one of your tumor suppressors that's not working properly, and it increases the risk. So that can be absolutely associated with breast cancer, uterine cancer, cervical, thyroid. So if that's already been established, that knowledge is power. And it's not necessarily something more you can do, but that's absolutely knowledge for them to pass on to their daughters and their sons.
Well, has it been established that she's just brought it up that, look, here's a link?
Oh, no. So where it is if that, that is a possibility, genetic counselor and working with a genetic counselor and looking at the possibility of Cowden's being an issue, and then specifically looking to see whether you or, ideally, at this point, it may not give you much more knowledge, but for her, it could be really important to understand, is she a carrier? And is she at increased risk? And then subsequently for her children and generations beyond to understand this. And I think, I'm going to say, this is my own personal opinion on this, is that it's very difficult. And I think one of the horrible things about germline mutations is this, many feel a guilt associated with it. I screwed my offspring. I gave them something. There's not anything that we can do to actually control this. I am terrified, honestly, that I have, not in my family, it's not up here because it's not as associated with thyroid, as BRCA mutation. And I'm pretty positive it's on my husband's side. And he comes from a long line of, don't worry about it. Don't worry about it. We don't share our health histories. We don't, there's very much a generational and a cultural divide in some of these things. And very much a stigma associated with, if I have a germline mutation, that I've done something wrong and I have done something to my future offspring. But I would argue there's nothing that we can do about it, but you're probably doing more harm by not sharing that information. And I'm still in the midst of working with my husband to get him to have germline testing, because they do have the history. He's got, his grandfather died of pancreatic. He has a aunt that has early onset colorectal cancer, 3 aunts in the family that have breast cancer diagnosed under the age of 50. Lots of other issues. All of it from where I'm standing is screaming at me that there is something in the germline here that we need to be aware of. His family wants nothing to do with it. And there is so much resistance because of this stigma culturally and generationally associated with it that I think makes it very difficult to have these conversations. And some people just don't want to know. I know for a fact his mother doesn't want to know. She wants nothing to do with it. I can't fix it. I can't control it. I'm better off being naive and being blissfully ignorant to what's going on. I want to know if my daughter has an increased risk of breast cancer, then we proactively can be looking for that and monitoring for it. I can't fix it, but I can give her the power that she doesn't become another statistic of early onset breast cancer that wasn't found in time or was ignored. But some of these things we can't do anything about, right? You know, Huntington's disease. I mean, that's not have anything to do with thyroid cancer, but when we don't have any type of intervention, we don't have any drug, we don't have anything that we can do, there are many that feel ignorance is bliss. I'd just rather not know. It's that question, you know, if you could ask and know when you were going to die, would you want to know or would you not want to know? And I think all of this is very individualized to people, and to each individual. The problem when you get into germline testing is you can't take it back, right? And if you have a germline mutation, the odds are you inherited it from mom or dad, and it was further upstream, and there's a really good chance that you're passing it down. When you do molecular testing, when you do those somatic testing, it's crap luck, but the chances are you can't blame mom and dad for it necessarily, and your kids can't blame you. It was a cell that went wrong, and it was only that one cell that led to something else. So genetic testing is that genie in the bottle. Once it's out, you can't put it back in. And that's where my absolute bias is that it should absolutely be done in consultation with a genetic counselor that can go through and translate some of this. What are the risks? What can you do? Is there prevention? Is there not? How do we utilize this information? I think it is so dangerous to get some of this information off of sending, you know, your spit in the mail and getting your genealogy back and, oh yeah, look, we found these different things, or we found these mutations and we have no idea what they mean. You know, all of us, we have millions, trillions of cells. We all have tons of mutations. Most of them aren't going to do anything. Our DNA is getting mutated and making mistakes all of the time. Most of them aren't going to end up being cancer, but those few that are, and how do we sift through that is critically important, and that's where you need a professional. It's just impossible. And I don't even know all the risk alleles. I don't know all the information. That's why I think it's so critical to work with an expert partner, geneticist, genetic counselor to kind of shift through and weed through that information.
From Robin, my 2 kids and I have a SMARCB1 tumor suppressor gene mutation, as did my mother, who died from ATC. My daughter has schwannomatosis, and I now have PTC. Have you seen this with SMRCB1 mutation?
No, I haven't personally, but the gene family, and this is where the difference between what I know physiologically and what I can say from the research setting and knowing what these different genes, proteins are responsible for versus really what the clinical link are very different. That is not something that's tested for on most of our panels. So that would not be something, you know, we're not looking for SMARCB mutations on ThyroSeq, at least as far as I know. So the prevalence and the role that it has in thyroid cancer is really still kind of not known definitively, because if you're at Harvard, if you're at CHOP, if you're at MD Anderson and you're getting your entire tumor sequenced, we're finding those things and we're reporting them. They may not be as rare as we think they are, but if you don't look, you don't find. And this is one of the challenges. And I think the big limitation is I talked about some of those commercial tests, when we're testing, why we're testing. We have a huge selection bias of what we know about because we only know about the things that we've looked for. We don't know about the things that we haven't looked for. And it's only now that we're starting to kind of open up Pandora's box to find those things that we hadn't historically looked for. So I've seen that mutation, I think, on some of our coming up, but I don't know of a targeted therapy or a definitive risk associated with it.
And we are at time, but there is one more question if you have time.
Sure.
This might be a difficult one. How
Oh,
do
boy.
we know we have found a good genetic counselor? Asked Jessica.
So, I mean, that's a good question. I don't know. I think one is to make sure they're trained in genetic counseling. And most of the genetic counselors, at least that I've seen, are associated with the cancer centers. Genetic counseling isn't necessarily like you go see your PCP for genetic counseling. It usually it's a pretty specialized field, and most commonly, from what I've seen at least, is associated with cancer centers. But there's additional credentialing that needs to take place for being a genetic counselor and being a certified genetic counselor. So there is specific training. It's not just I can decide I know enough about a molecular test and genetics that I can be your genetic counselor. So it definitely, I think, is something that should be done. And I think the best place to look is probably with a cancer center or talking with a PCP to try to get a referral to somebody that has the credentials in genetic counseling.
Okay. All right. Thank you so much for your time today, Dr. Franco.
Yeah.
And we will end it here.
Great.
Thank you.
Thanks, everyone.